Hello everyone, my name is Jessica Garcia. I'm a student at Texas State University, and today I will be presenting on the microbiome and the human body. So first of all, what is the microbiome? The microbiome is the collective genome of the microbiota living in and on us. It is composed of bacteria, archaea, protozoa, fungi, and viruses, and altogether, our microbiome provides 100 times more genes for us than our genome. We have so many areas inside and on our body that all provide a unique environment for our microbes. The three main areas that are the most studied and that we'll be covering are the gut, oral, and we'll be briefly touching on the skin microbiome. A way many of the bacteria are studied, classified, and identified is by using the 16S RNA gene. This is a ribosomal gene that has highly conserved regions with hypervariable regions. By amplifying and sequencing this region, many organisms have been identified that would not have been identified by culturing alone. It's important to note that individual microbiomes are very unique, and they are also unique to their location. So, for example, although the oral microbiome of all of us will be very different, there will be more similarities between your oral microbiome and my oral microbiome than with my oral microbiome and armpit microbiome, for example. There have been some noted geographical similarities, and geography has been seen to play more of a role in the gut microbiota, which could be due to similar climate and food people in an area are exposed to. The microbiome changes as we change. It has been found to be very responsive to lifestyle changes, and things that alter the microbiome include diet, medications, antibiotics, probiotics, which are life cultures of bacteria, prebiotics, which are food for the bacteria, and the natural aging process. A study with rats found that although these rats had very different diets throughout the study, and their initial microbiomes were very different, and their microbiomes throughout the study were also very different, at the end of their lives, they all had very similar microbiomes. In humans, it has been found that in elderly people, there seems to be less diversity of the microbes as we age. So, what does a healthy microbiome look like? Because of such a huge variance in the microbiome of healthy individuals, it's actually been hard to identify a healthy standard or a taxa that is seen in all healthy individuals. Initial microbiome research was looking for a core set, but they realized there was so much variance and no core set could be found. Instead of finding a core set of microbes, they found that there were different microbes that performed a set of important functions in different areas of the body. This led to a shift in thought that a healthy microbiome contains a functional core instead of a core set. And this functional core, or things that make this functional core healthy, are things like resistance to stress, the ability to recover after a stressful event, diversity, balance, and ratios between certain microbes and in certain areas. So, this is an illustration from the paper you guys read, and it's depicting how in three different people, the microbial taxa that is prevalent can vary. However, there are core enzymes, metabolites, and pathways that make up the functional core and are important at the different body sites. All right, so the first area we will be discussing is the gut microbiome, which is the most diverse and studied microbiome location. There are many bugs that live in the gut, and some of the potential fungal colonizers include Candida, Saccharomyces, Aspergillus, Cryptococcus, and lots more. Antibiotic treatment has been found to promote the growth of Candida by killing or reducing good bacteria in the gut and allowing for growth and sometimes overgrowth of Candida, which can have negative effects. The bacterial colonizers have been found to be predominantly belonging to the Bacteroides and Firmicutes phyla, and there seems to be a correlation between the ratio of bacteria in these two phyla and metabolic conditions such as obesity, which we will be touching on shortly. So, the development of the the gut microbiome. The gut is thought to be sterile in the womb, and birth is the starting point of colonization. The first colonizers are acquired from the environment and maternal contact. That are born through vaginal birth have higher amounts of the Lactobacillus and Bifidobacterium species. This is because these bacteria are found inside the vaginal canal. Babies born through cesarean section have been found to have higher amounts of Staphylococcus in their gut microbiome, and this is because Staphylococcus is found on the skin. When we come into the world, our first source of food is usually breast milk, which is very important. Not only is it full of nutrients and antibodies, it's also a probiotic, which provides new bacterial colonizers to the gut. These colonizers help break down simple sugars that we're eating from the milk. As we grow, we are also introduced to new foods, and these foods introduce new bacteria to us. These bacteria come with new functions which help us break down more sugars or more complex sugars and synthesize vitamins. Things can also have negative effects on the gut microbiome, such as maternal health status, malnutrition, premature birth, milk quality, and antibiotic use. Studies have shown that obese mothers have less diverse bacteria in their milk, which can have a negative effect on the microbiome development of the infant. Antibiotic use has been linked to asthma, childhood obesity, allergies, autism, and eczema. It's important to know that when using antibiotics, we're not just killing the bad bacteria, we're also killing the good bacteria. So they must be used as instructed by doctors and with caution. Food and the microbiome. The gut is very responsive to dietary changes and influenced also by our long term habits. Probiotics are the live cultures of bacteria that we can eat, which can be in supplements, yogurt, kombucha, fermented foods. Prebiotics are the food for the bacteria. So these can be also in supplements. It could be our fiber rich foods that we're eating, such as fruits and vegetables. And the postbiotics are the bacterial metabolites, which can be taken as supplements. Fiber deficiency has been found to be connected to an increase in mucus degrading bacteria, which can be negative for us, and also an increased susceptibility to Citrobacter, which has been found in UTI infections. Animal-based and plant-based diets are very different, and they also have different effects on our microbiome. An animal-based diet, for the most part, consists of a high-fat intake. Not always, but with a high-fat intake, you are increasing bile acids and bile-resistant tolerant microbes. Also, there is usually, not always, but usually, lower intakes of carbs and this leads to lower levels of products of carb fermentation. There is also higher concentration of products of amino acid fermentation, and with a plant-based diet, you have more carbohydrate consumption, so you have higher levels of products of carb fermentation with that higher fiber intake. That is also more prebiotics for the bacteria, and this affects the diversity of the Clostridium cluster XIVA. And there are studies looking into how this affects our health. So the bacteria produce different metabolites, and some of these metabolites are short chain fatty acids, which include butyrate, propionate, and acetate. These are produced by the ingestion of our dietary fiber, which is then fermented by the bacteria to produce these short chain fatty acids, which are then used in our body for energy, countering inflammation, maintaining gut homeostasis, and in our microglia, which has been found to connect our gut brain connection or our gut brain connection. However, these can also be toxic at high levels. Bacteria also produce bile acids, neuropeptides, neurotransmitters such as dopamine, acetylcholine, and serotonin, and polyamines such as putrescine and spermine, which have anti-inflammatory effects. The immune system and the gut. So, our immune system is educated and matured by our bacteria, so we have a lot to be thankful for to them. LPS stands for lipopolysaccharide, and this is produced by bacteria and believed to cause low grade inflammation and is mediated by inflammatory cytokines. Studies with germ free mice have found that they have abnormal immune systems, and also with having an absence of a microbiome, they have also an absence of lymph nodes or underdeveloped lymph nodes that are seen. And we know that lymph nodes are important for the immune system. So, different metabolic conditions such as diabetes or obesity have different microbiomes associated with it. Diabetes is characterized by altered microbiota, inflammation, and a gut barrier disruption. Obesity is characterized by a different F to B ratio, and it is seen throughout various people with obesity that there is a high F to B ratio. There's also seen to be less diversity with the bacteria and dysbiosis. L. reuteri has been seen to be increased and M. smithii depleted. And studies are still looking for consistency between people with obesity and the bacteria that are present in their gut, and also the metabolites that they produce, as well as the functions that are or are not being um, done by these bacteria. Moving on to the oral microbiome, this is the second most complex microbiome on our body. There are over 1,000 species that have been found in the mouth, and it's estimated that about half of the bacteria still remain to be cultured or discovered. Viruses, protozoa, fungi, and archaea also are living in our mouth, and it's found that a negative interaction between the host and the microbe results in a disease state. So, the core microbiome functions in the mouth include simple carb breakdown and regulation of pH. This is very important in our mouth. Geographical differences, as I had mentioned before, have an impact in the microbes that develop in the mouth. This has been thought to be related to the food and the diet that is available in those different areas. Things such as fermentable carbohydrates have an effect on the bacteria and what they do in the mouth. The bacterial community is mostly dominated by firmicutes, bacteroidetes, proteobacteria, actinobacteria, spirochetes, and fusobacteria. Biofilms are very interesting and are seen in the mouth as well as in other locations where we have microbes. But looking in the mouth, what happens here is the bacteria attach and they begin to grow and they grow a film over themselves and other bacteria around them. And inside these films, the bacteria grow and they multiply. They benefit from each other's different genomes that they have. They share enzymes, they share metabolites, they share food, and they grow together as a community. Once they want to, they detach and grow somewhere else and grow another film around them. And these films can be pretty hard to get rid of. In our mouth, we have few obligate aerobes and many facultative anaerobes. Nitrate metabolism and cardiovascular health. So, around a quarter of our ingested nitrate is returned to the mouth, and here the oral bacteria actually reduce it to nitrite for us, and the nitrite is taken up into the bloodstream and converted into nitric oxide. And nitric oxide is essential for our vascular health. It helps keep our blood vessels flexible and has an antihypertensive effect. Dental caries or tooth decay, which then develops to a cavity or a deep carie, is caused by oral bacteria fermenting our carbs and producing an acid. This is made worse by ingesting high levels of carbs and increasing the frequency of the acid production. If you have a high frequency of the acid production, this decreases the ability of our saliva to be able to buffer the mouth. And if you have a decrease in the buffering ability, your pH is reduced, and this favors the acidic species such as Streptococcus mutans and Lactobacilli, and that is how the caries are formed in the tooth, which then develop into the cavities. Periodontitis is inappropriate inflammatory reaction to the normal microbiota. This can be made worse by the presence of disease-associated species, but it's not necessarily caused by these. And these are listed right here, and I've tried to pronounce them, and I failed miserably. So here they are. What happens here is a loss of attachment between the gingiva and the teeth, and a periodontal pocket fills with anaerobic bacteria, which is very bad, and this just keeps growing and the condition can worsen. A thing that it causes someone to be susceptible to this is tobacco use, so that's important to note and just keep in mind. You don't want this. Endodontic infections. So here we find anaerobic proteolytic bacteria, and persistent infections include enterococci. And it's important to note that enterococci are not normally found in a healthy mouth. And patients that are going into for an endodontic treatment or anything like that are usually advised to avoid consuming foods that are known to be colonized by enterococci, which include cheese, dairy, and fermented meats. And this is because you don't want to be introducing this bacteria and potentially cause an endodontic infection or continue with an infection. Gingivitis. So gingivitis is caused by plaque continuously forming on the teeth and then causing inflammation. So what happens here is the primary colonizers, which are gram-positive and facultative anaerobes, which can include streptococci and actinomyces species, as well as many others, start colonizing on the teeth and forming the plaque. And then this turns into mature plaque, which contains anaerobic gram-negative bacteria such as Fusobacterium, Treponema, and members of the phylum Synergestes. So, with this, oral hygiene is very important, and with this condition, it can be reversible. So, with regular teeth cleaning, our plaque is kept in an immature state, but with irregular teeth cleaning, this increases the gram-negative species and endotoxins and enzymes that are produced, which leads to a increase in the condition. So you can definitely go back and forth between having it or not having it. It can worsen to a point where it gets very bad. And it can be irreversible, but at the beginning stages, it can be reversible by maintaining a good oral hygiene. And it's best to not let it get to that level, not let your plaque mature, keep it in an immature state. Moving on to our skin, which is a very different and external environment. This consists of dry, moist, and sebaceous locations. The richest site would be the volar forearm. The least rich is the retroauricular crease. Most even site is the popliteal fossa, and less even would be our back and the web space between our toes. On our skin, there is found to be symmetry within the sites since we are symmetrical, and even though these sites would be pretty far from each other, speaking in bacterial terms, our arms, so our right forearm and our left forearm, although they are on opposite sides of the body, they will contain very similar microbes. And that goes all throughout our body with the symmetrical locations. So with atopic dermatitis, on the skin, there's usually highly diverse, rich, and even in terms of bacteria compared to other sites. Here, the skin inflammation actually promotes the growth of certain bacteria, which would be the S. aureus. And if there are breaks in the skin, the bacteria can get in there and make the condition a lot worse. Psoriasis is something that is not always caused by bacteria, but can be triggered by a bacterial infection as well. And the bacteria here would be Streptococcus pyogenes. The main sites would be the umbilici, gluteal crease, occipits, elbows, and the knees. This, like I said, can be triggered by a bacterial infection and can be treated with antibiotics or other medications. Acne is also something that can be caused by bacteria or triggered by bacteria or made worse by bacteria, not always started off with bacteria. But what happens here is we get strains of Propionobacterium that are usually associated with sites that have acne, and these bacteria are lipophilic anaerobes that like to live at the sebaceous sites and around these glands, and they feed off of the fat and really thrive there. So, in summary, we have seen that we have a huge genome, 100 times more genes found in these bacteria and viruses. And I didn't really cover viruses, but in all these microbes living in and on us, way bigger genome provided by them than by us. We've seen that our lifestyle can have a huge impact in what kind of bacteria are living in and on us, as well as heredity or how we came into the world. The bacteria that are living on us or in us can have positive and negative effects on our health. And there's so much research going into this area. It's a growing field. like I mentioned with the mouth, there's still so many species that are yet to be discovered, and it's really amazing what has been found that these bacteria are doing in the different sites in our body. So if you guys have any questions or have heard of any research that's going on in the microbiome world, please let me know, and thank you so much for listening.